Welcome to this lesson. We are going to focus on uses of lenses in optical devices. Now, due to their ability to converge and diverge light rays, lenses are widely used in optical devices. The devices include 1. The human eye, 2. We have simple microscope, 3. We have compound microscope, and then we have the camera. Now, to start with, we are going to look at the human eye. So this is a natural optical instrument, and you can see the main parts. We have the cornea, we have the pupil, we have the iris, we have the sclerotic layer or white of the eye, choroid layer, which is also black. So if you look at your eye, the white bit is just sclerotic layer and the black bit is choroid layer. Then you have the retina just below the choroid layer there. We have got fovea. Biologists will call it the fovea centralis. Then we also have aqueous humor. So then we also have crystalline lens. We have the ciliary muscles, which are just attached to the crystalline lens through suspensory ligaments. Now, let's look at these parts and their functions one by one. We have got the hard shell that enters. We have the sclerotic layer. So this is just the hard shell that encloses the eye, and it is white. The front part is transparent and spherical, known as cornea. Most bending of light entering the eye occurs at the cornea. So it means a lot of refraction takes place at this point. Then we also have the aqueous humor. So this is just a clear liquid between the cornea, as you can see, and the lens. So it helps maintain the shape of the eye. Then we also have the iris. This is just the coloring of the eye. It has pupil, which regulates the amount of light entering the eye, meaning if light is too much, it becomes small. If the light is of low intensity, then it enlarges. Then we have got the crystalline lens. So this is just the lens that is capable of, 1, it refracts the light, and also 2, it is capable of changing its focal length by the help of ciliary muscles. That enables one to see far objects clearly as well as near objects clearly for a normal eye. The vitreous humor is just a transparent matrix liquid between the lens and the retina. So that's where it also fills the eye, and so that goes without saying, it maintains the shape of the eye. Then we have the retina. So let's look at where the retina is. When you look at where the retina is, you realize that it is where the image is formed and made of cells that are light sensitive. If you read from other books, you'll realize that this is where we have the rods and cones. So the cones are capable of forming image at light of high intensity, and the rods uses light of low intensity. Now we have the fovea. Fovea is the central part of the retina that exhibits best details, and color vision takes place at that point. So if you have problem in differentiating colors, then it means your fovea has a problem. Blind spot contains cells that are not light sensitive. Then ciliary muscles, these are just muscles that support the lens and control the shape of the lens by contracting or relaxing. So in relaxing, the muscles enable the lens to increase, hence focal distance, hence focus distant object. Then in contraction, the muscles reduce tensions in the lens to increase its focal length, thus focus near object. So this process is known as accommodation of the eye. So the near point is just the closest point which a normal eye can focus, and that is normally around 25 centimeters for a normal person. But the far point is the farthest point that a normal human eye can see. So it may differ in different people. Now, it is also important we talk about the defects of the eye. The first one is short-sightedness, also called myopia. So this is whereby a person can only clearly see near objects. So rays of near objects are focused on the retina, but those for distant objects are focused in front of the retina. What causes this is short focal length of the human lens, eye lens, and also long eyeball. And it is corrected by diverging lenses as shown in that diagram. The second defect of the eye is called long-sightedness, so also hypermetropia. So it can see, this is where one can see distant object but not near ones. The image of near objects are formed behind the retina. So causes are too long focal length of the eye, too short eyeball. So you can see it in that diagram. This person is able to see a distant object, but near ones are focused behind the retina. Now, it is corrected using the converging lenses as shown in this diagram. The second application or use of the lens is the camera. We have now just talked of the human eye. So cameras, lenses that focus light from objects to form an image of the object on the film. Focusing is done by adjusting the distance between the lens and the film. Diaphragm controls the amount of light entering the eye. The shutter allows light to reach the film only for a precise period when camera is operated. The inside is blackened to absorb light. So, what are some of the similarities between the eye and the camera? You'll realize that eye has got crystalline lens, convex lens. Camera also has the convex lens. The eye has got choroid layer which is black. The box of the camera is also painted black on the inside. The retina, where images are formed on the eye. Camera also has light sensitive film where images are formed. Iris controls the amount of light entering the eye. Diaphragm controls the amount of light entering the camera. So we conclude that there are similarities between the human eye and the camera. What about the differences? In as much as they are similar, there are also differences between the eye and the camera. One is that the eye has got crystalline lens which has variable which varies its focal length, while the camera, the lenses have got fixed focal length. Now, eye has got constant image distance. The distance between the crystalline lens and the retina does not change, while the camera varies the image distance depending on the distance of the object being focused. Then the eye can take constantly changing pictures. Then for the camera, only one photograph can be taken at a time. So, number 3, we have got the simple microscope. Simple microscope is just a magnifying glass. So when the object is placed between a convex lens and its principal focus, the image formed is virtual, erect, and magnified. That is the concept which it uses, and you can do it as shown. Then another application or use of the lenses is the compound microscope. So there are two cases under which converging lenses can produce magnified images. That is, when the object is between f and 2f. In that case, we know that a magnified real image will be formed beyond 2f. And then, when the object is between the lens and f. So a compound microscope combines the two cases above. So it consists of converging lenses of short focal length. The focal length next to the object is called objective lens, and one next to the eye is called the eyepiece or ocular lens. The objective lens is of short focal length. Now the eyepiece is also of short focal length but longer than that of objective lens. A compound microscope overcomes the limitations of simple microscope by use of objective lenses with many lenses and eyepiece with more than one lens. Now, total magnification produced by the compound microscope is given by this formula: VE over FE minus 1 into V naught over F naught minus 1, where V naught is the image distance from I, if you check that diagram, and then VE is the image distance from I, again, if you check on the diagram. Just on the slide, we have concluded. Now, you can take time and do this question. So, thank you for watching.